Hi, I'm Joseph Gleason from UCSD and Radiogenomics Institute to introduce our recently published paper in Nature entitled Cell Type Resolved Mosaicism Reveals Clonal Dynamics of the Human Forebrain. This work was mainly performed by Cheng-Luk Chung and Xiaoshu Yang, who are the first authors of this study. The human brain is very complex, especially compared with other species like rodents. Humans have a very large and wrinkled cortex, depicted here artistically. This expansion likely supports human higher cognitive functions. But how is this complex structure formed, and what sorts of brain diseases can result from impaired development? The human cortex consists of billions of neurons that are interconnected through synapses to achieve brain function. Pashko Rakesh proposed that cells in the cortex or forebrain derive exclusively from progenitors in the ventricular zone. It turned out later that essentially all excitatory neurons, abbreviated EXN, originate in this location. But there's another type of cell in the cortex called inhibitory neurons that release a transmitter that quiets the brain circuits called GABA. In the 1990s, Rubinstein and others proposed the source of these GABAergic cortical interneurons are the ganglionic eminence, and they produce these cells depicted here in red. The key experiment was in mice missing the canonical inhibitory factor DLX1 and 2, and those mice showed no inhibitory neurons in the cortex. This model has persisted for more than 20 years. However, in recent studies, cultured human fetal cortical progenitor cells were able to generate not only excitatory neurons, but also inhibitory neurons, as depicted at right. And this suggests that there could be a second potential source of inhibitory neurons in humans, i.e. coming from the same dorsal cortical progenitor cells as the excitatory cells. We call these cells DINs, D-I-N-S. Something similar had been suggested by Rakesh's lab about 10 years ago. Studying in vitro monkey brain slices, they found evidence that some interneurons, here in blue, might derive from the ganglionic eminence, depicted as possible scenario A, or might come from dorsal progenitor cells, as depicted in scenario B and C, where these red cells give rise to the blue cells. However, whether this happened in human in vivo was untested.
Hi, I'm Dr. Cheng-Luk Chung, a postdoc in Gleason Lab. As Dr. Gleason mentioned, there has been no in vivo evidence of DINs in humans yet. If DINs exist, this can be one of missing points in mice and organoid models. Also, all studies modeling human cortex should take this lineage into account. However, it has been challenging to observe the cell lineage dynamics directly accessing to mature human brains. But here we used mosaic variants as lineage markers to answer whether DINs exist or not in human cortex. We solved this issue by using mosaic variants. During embryonic development, mitotic cells acquire genetic variants called mosaic variants in their genome by various intrinsic or extrinsic factors, and these variants are transmitted to daughter cells, basically labeling cell lineages. For this reason, mosaic variants can be used as natural lineage markers or lineage barcodes. So utilizing mosaic variants is powerful for observing human brain development process within in vivo or in vitro models, because we can just directly access to the human brain and infer human brain development process without using models. So let me introduce how we tested the lineage relationships between different cell types using mosaic variants. First, we obtained donor's brain within 24 hours of post-mortem interval. Then we carried out the tissue collection from many different cortical regions. A subset of the punches was subjected to bulk whole genome sequencing, and we made a mosaic variant candidate list. In the next step, we isolated different neuronal cell types using a sorting protocol called methanol-fixed nuclei sorting. We were successfully able to isolate high-purity DLX1 positive, TBR1 positive, and CUPTF2 positive nuclear pools, which represent pan-inhibitory neurons, excitatory neurons, and caudal ganglionic eminence derived inhibitory neurons, respectively. Then, using the mosaic variant candidate list prepared by whole genome sequencing, we measured the early fractions of the listed variants in various brain cell types in various tissue punches. This early fraction matrix was used for downstream analysis. Here are some examples of mosaic variant distribution by early fraction values. We observed dozens of mosaic variants exclusively specific or enriched in CUPTF2 positive inhibitory neurons, but not in TBR1 or DLX1 positive neurons, suggesting distinct origin of CUPTF2 positive neurons. Especially they were not confined to the specific cortical regions, but were widely distributed across the cortex. This result implies this CUPTF2 positive lineage was ventrally derived inhibitory neurons, and it is tangentially migrated and dispersed widely to the cortex. What surprised us was there were more than 10 variants like this variant, which were very exclusively present in only the local area and only in TBR1 and DLX1 positive neurons, not in CUPTF2 positive neurons. These results suggest that the majority of DLX1 positive inhibitory neurons and TBR1 positive excitatory neurons were derived from the same local dorsal ancestors. Next, to investigate the genomics relationships considering multiple mosaic variants, I generated heat map plots with early fraction matrix from 2 donors and 4 different hemispheres. Each sidebar indicates hemisphere, lobe, and the cell type identity information. CUPTF2 positive nuclear pools were exclusively clustered by themselves, meaning that CUPTF2 positive nuclear pools are genetically similar across lobes but different from DLX1 or TBR1 positive nuclear pools in the same lobe. Meanwhile, DLX1 nuclear pools were clustered with TBR1 positive nuclear pools within the same lobe, meaning that the majority of the inhibitory neurons and excitatory neurons are genetically close in the same punches. Taken together, these clustering results implies a significant number of DLX1 positive inhibitory neurons must be dorsally derived inhibitory neurons, whereas CUPTF2 positive neurons derive from a very distinct origin and widely dispersed across the cortex.
Hi, I'm Dr. Xiaoxu Yang. Finally, we seek to validate dorsally derived inhibitory neurons at a single cell level. Single neurons were isolated from frontal cortex and temporal cortex, and simultaneous DNA and RNA sequencing is performed from the same nuclei with the BioScribe Resolvome technology. Genotypes were determined from single nuclei DNA, and cell typing were performed in single nuclei RNA. This double-ranked plot displays genotype and cell type of 118 nuclei. Each column represents a cell, and each row represents genotype of each variant in each of the cells. If a variant is positively detected in a given cell, it is marked with black. Cell type was determined by a UMAP clustering from the single nuclei RNA-seq. Among the 118 cells, 33 were inhibitory neurons and labeled with red. 85 were excitatory neurons labeled with green. Notably, some inhibitory neurons were observed to share local variants with excitatory neurons, which implies dorsally derived inhibitory neurons.
Variants shared between inhibitory and excitatory neurons within each lobe are further colored. For example, those inhibitory neurons sharing the same variants with the excitatory neurons within the same lobe are labeled in dark red as inhibitory neuron 2, whereas the others are labeled as inhibitory neuron 1. We observe 16 inhibitory neuron 2 here. The chance of observing such great number of inhibitory neurons sharing the same variants with the excitatory neurons in the same lobe is very low according to a permutation test. Thus, this experiment strongly suggests the dorsal origin of inhibitory neurons that shares the same common ancestor with excitatory neurons within the same lobe.
In summary, in the postmortem human brain, we identified cortical inhibitory neurons sharing local origin with excitatory neurons using mosaic variant barcoding. This is the first in vivo evidence supporting the existence of dorsally derived radially migrated cortical inhibitory neurons in humans. Meanwhile, CUPTF2 positive neurons showed very distinct mosaic variant composition and distinct distribution patterns supporting a ventral origin of inhibitory neurons.
Several conclusions emerge from these studies. First, mosaic variant barcode analysis can reveal cellular origins across the neurotypical mature human forebrain. Second, in vivo evidence for such a dorsal source of cortical inhibitory neurons is revealed. Several future questions emerge. When and where are dorsal inhibitory neurons born? What are the cellular identities of these dorsal inhibitory neurons? And what are the roles for these dorsal inhibitory neurons in the cortex? We'd like to acknowledge all of our amazing collaborators, the members of the Gleason Laboratory listed here, UCSD collaborators, and our funding sources. Thank you so much.